Welcome to the Australian Biocommons webinar series. Through these sessions, we hope to share useful information about the latest digital techniques, data and projects. We offer this platform to local and international peers to support Australian bioscientists to deliver their best environmental, agricultural and medical research. My name's Christina Hall and I'm the Australian Biocommons Training and Communications Manager. We particularly appreciate those of you joining us live today, and you'll have the opportunity to ask our speaker questions via the Q&A function on your dashboard, and these will be addressed at the end of the presentation. This session will also be recorded if you would like to revisit it in the future, and you'll find it on our YouTube channel, along with recordings of previous webinars and workshops. We also hope you'll keep in touch to hear about future webinars via the channels listed on the screen. Before we start today, we'd like to acknowledge the traditional owners of the lands on which we work and pay our respects to the elders past, present and emerging. With us today is Professor David Cantrill. David is the Executive Director of Science at the Royal Botanic Gardens of Victoria. He's an honorary professor at the University of Melbourne in the School of Biosciences, and at the moment he's heading up the Royal Botanic Gardens Operations Working Group in response to the pandemic. So we're very lucky that he's found the time to be with us today. David's research career started at the University of Melbourne with undergraduate studies in botany and geology and a PhD from the School of Botany. He's travelled to Antarctica 7 times with the British Antarctic Survey as a paleobotanist, and also worked at the Museum of Natural History in Stockholm. He returned to Melbourne in 2006 as the Chief Botanist and Director of the National Herbarium of Victoria at the Royal Botanic Gardens. David has authored over 140 publications, including a recent co-authored book, The Vegetation of Antarctica Through Geological Time. He's very active in the international science community with many editorial and committee positions of prestigious journals and societies. And today we'll hear about his role as the chair of the steering committee of the Genomics for Australian Plants. So with that, I'll hand straight over to David.
Great, thanks for that, Christina. That's my great pleasure to be here today, and as you will have heard from the molecular biology is not really my thing, so it's a bit curious that I find myself sitting here today talking to people about a very exciting project that is running at the moment called Genomics for Australian Plants. What I want to do today is I want to run through a little bit of background first on how our community works, and how we've come to this point in time where we've got a large national project being supported by Bioplatforms Australia, various philanthropic trusts, and also what I refer to as the plant collections community. So Genomics for Australian Plants is a consortium-based project to build capacity across our community, as well as to upskill, but also to develop a whole lot of genomic resources that we want to use or we think are going to be valuable for us. But just as background to that, I just want to look at where we've come from. So you can see a lovely image here of a whole pile of pressed material from the 1800s. And this is really what we're dealing with day to day within our community. We're using this material for a whole range of purposes. This particular box is one that we acquired a few years ago, and it comes from a collection made by a French botanist in the 1800s, and it contains a large number of type materials. So this material goes back to the 1800s, and some of our oldest specimens go back to the 1500s. And that's a resource for the whole research community that's available through a number of herbaria. If we have a look at what a typical specimen looks like, you'll see here there's the plant material, which still contains DNA, and a whole lot of other information. And these specimens are our day-to-day research tools for the community. So the herbarium and plant collection community has a peak body that represents those institutions, the Council of Heads of Australasian Herbaria. And that organisation has a long history of collaborative projects It's quite unusual in an international context that we've been able to build these projects and collaborate at a national or a continental scale. And just as an example, I've listed 4 sort of big projects that have been running, and I'll just run through them and how they're benefiting the community, because it provides context for the GAP project, the genomics for Australian plants. So if we look at these specimens, there's a whole lot of information that's attached to them. And you'll see here I've highlighted names. So there are a number of names on this particular sheet. And so the question then is which is the correct name? You know, is it this one at the top or is it the one at the bottom? And what do we accept as a community as the valid concept? So there was a project that was begun, probably it's close on 20 years, it might be a bit longer than that now, called the Australian Plant Name Index, and that was to gather all these names together. And in Australia for vascular plants, there are over 75,000 names that have been used for our flora, which is quite different to how many species we actually recognise. So one of the reasons for that is when material was collected in the 1800s and plants were taken back to Europe, they were distributed across the European continent, and people on different parts of the continent recognised them as new species to science and described them and gave them names. So often more than one species, or each species had more than one name attached to it. And our challenge has been to disentangle that problem over time. And so we've discovered about 75,000 names, but there are approximately only 25,000 species concept recognised. And this project has been a fully collaborative one where there are working groups that are supported by the institutions that are gathering those names together and putting them into concepts, and then nationally the community agreeing what the correct name is and what the concept is and what names are attached to that. And this is the only continent on the planet where this approach is being used. Often they're single institutional views or single jurisdiction views that are in conflict with other views of the taxonomic framework. And that taxonomic framework is critical because taxonomy underpins all biological sciences. So the other bit of information that you see on these sheets is they've got information about location, collector and date, and where those things have been and who made those collections. And that information is important for disentangling the names issue, because we can understand who's looked at specimens and when they might have looked at them. And that helps us understand the concepts that they've created in terms of the names. And so we started a program called Australia's Virtual Herbaria first, but then became the Australasian Virtual Herbaria, where we're capturing this information. We're capturing this information for our purposes in terms of discovering and documenting the diversity in the Australian, or now Australasian, region. And so the Australia's Virtual Herbaria is a great record of collaboration across all the herbaria across the country. It brought the community together. It made us recognise a whole lot of things that we need to do if we're to make these sorts of projects successful. So things like having common standards, agreed data fields, and all those sorts of things that we use in many areas of research. And to date, there's about 22 contributing herbaria with 6.5 million records. And so while that program was to support taxonomic and research and biodiversity discovery research, by making it accessible and freely available, we've enabled a whole range of new research to happen. And that's sort of one of the exciting things of doing these national collaborative projects. It also is a big benefit to the individual research institutions as well. So for example, you'll see on that bottom bullet point, we've had 190 million records downloaded from our database in 265,000 download events in probably the last 8 years. Yet we only have 900,000 records in the database. So that data is getting reused over and over again. And that's another important aspect, I think, of these sorts of programs, that whereby we create data sets, but making them accessible enables us to get data reuse happening. But you've got to design the programs in the right sort of way. And I guess, you know, some of the things that we hadn't anticipated, which are just, you know, standard research tools now, are things like the use of this data for species distribution models, and a whole range of other things that have sprung out of us creating this resource. So the question then is, you know, what else can we do with Herbaria records, and why are they important? So having captured a lot of the label information in Australasian's virtual herbaria, the next step that we're looking at is, you know, the digitisation, or the availability of those images. And again, this is probably a little bit self-centred, because we recognised if the images were available, we mightn't have to send out so many loans of material and risk damage or loss of specimens. But importantly, some specimens are more important than others. And so when species are described, for those names to be valid, they need a specimen that's tied to them, and that specimen acts as the reference or the gold standard against that name. And so those specimens are particularly important. And so another project that we've been involved in is the Global Plants Project, or the Global Plants Initiative project. And this was a $70 million US dollar program funded by the Andrew Mellon Foundation. And it started out as a project to virtually repatriate African specimens, because it was enormously expensive for African researchers to get to the European institutions that held these specimens. And so this was a way of making the specimens available for those researchers. And Global Plants now has around about 4 million images in them of type specimens across the globe. And we've contributed in Australia probably about 100,000 of those images, because not surprisingly, a lot of our type material is held in European institutions as well, as, you know, in the same way that the African or the South American countries have got an issue there. The interesting thing about Global Plants, again in that, you know, collaborative space, is that while those images were created for taxonomic and biodiversity purposes, they've had a whole range of other unexpected uses. So there are programs that are using these images to look at historical events, to look at where explorers went. There's been education tools that have been developed around this program that's been pushed into the high school. So it's sort of a reuse of this digital resource that's happened. And we continue to discover these unusual specimens in our collection. So the collection here in Melbourne, for example, we have about 600 or so thousand specimens from a global collection, about a third of our collection, and most of that is uncurated, and we're currently working our way through it. And as we do, we find material and we make that available to the community. And so you can see that on these sheets, there's a thing that says type, and so that's a reference standard specimen for a Amorphophallus. Of course, there are a whole lot of other data streams that are attached to and can be attached to these specimens, and some of these are available and some of them are not. So here we've got the specimen. There are images of specimens before they were collected that give information on habit and colour and things that don't preserve. There are, of course, preparations that are made from specimens. So, for example, slides of pollen or cuticle or other parts of the specimen that have been made for research purposes. and even SEM stubs and, again, pollen from the specimens. And I guess the important thing is that all this data ties back to a specimen that has a name. And if the name changes, we can then flow that through to all these other data streams. So that's sort of one of the fundamental, I think, value propositions that rest in these specimens. And you can see here I've written down morphology and traits is actually a interesting area of research that's happening at the moment. The recent bushfires that we've had in southeastern Australia, we've been bemoaning the fact that we haven't got a national plant traits database, because that traits database would enable us to predict which plants might be in more trouble than others and which plants we should be paying attention to. But you can sort of see how associating these data streams, we're building something that opens up new possibilities. So having said that, how does that relate to this talk, I guess? And I guess some of the things that have been happening in this area is the Global Genome Biodiversity Network, and I'm sitting on that committee at the moment, and it's developed the data standards that allow us to tie genetic data back to specimens, because they've recognised the importance of having specimens backed up to the DNA sequence, because it means that the currency of the names of those sequences, or the names of the organisms, can be kept up to date. And that's a really important point. But I think also by tying it back to the specimens, we then open up the possibilities for looking at genetic information in a spatial way as well, because if all these dots on the map represent specimens and we had genetic information, we could start to do some really interesting things. Now, whether that's something for our community to develop, or whether that's for someone else, is an open question, but you should be aware of the Atlas of Living Australia. This is another bit of NCRIS infrastructure, and that provides these sorts of analysis portals of these large biodiversity data sets. It's a data aggregator, so it's not a data custodian, so we or other institutions or other data holders provide the information into the Atlas, and the Atlas creates a bit of infrastructure that you can then look and analyse this information. And so what I would say is that taxonomy underpins all areas of biological science. It's critical to a range of other areas. If we don't know what the organism is that we're working on, or we don't know what the current name it is, a lot of that data doesn't have as much value. We have a strong use of this species stuff in terms of evolutionary relationships. So it's all very well to document a new species and demonstrate it's different, but we need to put it into a phylogenetic context, because that gives us a predictive framework for a range of studies. And the other critical point I think here is that voucher-based data sets enable greater data currency, because the taxonomy that underpins those data sets can be updated if taxonomy changes. And I guess, you know, to illustrate that, if you have a whole lot of bird observations that says, I've seen a, you know, speckled something or other, and the taxonomists come along, they do some genetic studies or whatever, they say, no, this is really two species. All of a sudden, what happens to that observational data? You've got no way of separating out where the two species are. And so, unlike voucher-based data, observational data sets degrade through time, whereas the voucher-based data sets actually get better through time. And so we often view Herbaria as research tools that actually improve through time by adding data streams to them. And one of the data streams that we want to add is through the Genomics for Australian plants. So that's a bit of a preamble on our community and our background. I just want to have a quick look at some of the international things that are happening, and then we'll move on to the GAP program. So you will have seen, or some of you will have seen, the UN report that came out last year that said there are more than 1 million species threatened with extinction, which is a staggering number, and just highlights the impact we're having on the planet. And while a lot of conservation focus is on animals, plants are in just as much trouble as animals in many respects. And Kew Gardens has been preparing reports over years. This one's a little bit dated now and they haven't done an update on this one, but they prepared a report, State of the World's Plants, which looked at conservation status, you know, how our documentation's going on them, and a whole raft of other things. It's a really interesting report, and they did one in 16 and one in 17, and then they moved on to look at fungi, which are another group of plant, or another group of organisms that our community works on. And if we look at the Kew report, they've got a section in there on genomics and what's currently known about, or you know, what genomes have been assembled for plants. And I've had a look at that data set and I've updated it a little bit. Certainly the number of genomes has accelerated. You can see the graph in the bottom left-hand corner that shows the rate of production of plant genomes. But it's very interesting when you look at the coverage across the phylogenetic tree of life. And so this data just relates to flowering plants, but if you look at it, we've now got a genome in 64% of the orders. We've got 22% of the families covered. So there's one genome in 22% of the 413 families. But when you get down to lower levels, there's only 2% of genera covered and a paltry 0.14% of species covered. The interesting thing is that this is not representative across the tree of life. It's concentrated in the food, crop and wild relatives part of the tree, and only 2.5% of those genomes that are available are for plants that aren't utilised. So we're really missing a big bit of the biodiversity, and we're really missing the, you know, the conservation end of the spectrum. So in Australia, the plant taxonomic community's been working, well, not just plant, it's the plant and animals have been working on developing a decadal plan, which is now out. And that decadal plan looks at the challenges ahead for the country in terms of documenting and discovering that biodiversity before it goes extinct. And if we can do that, we might be then in a position where we can start looking at how we mitigate against extinction. That report's got a lot of interesting information in it. And I guess some of the things that we're seeing are the information that's coming out of the eDNA studies, which point to a whole lot of what we refer to as dark matter, biodiversity dark matter, whole branches to the tree of life that have been invisible. And this is particularly important in the fungal space. So as an example, the microbiome project that was run, we pulled all the fungal sequences out of that and matched them to fungal sequences that were known from specimens. And there were around about 50,000 fungal taxonomic units, depending on how you cut the DNA data, yet only about 8,000 of those could be matched back to known and described species. So it sort of illustrates the depth of our lack of knowledge of certain groups. So the DNA world really offers us some great opportunities in that space. And this decadal plan recognises that. And one of the strategic actions within it is to build a curated and vouchered reference library of DNA sequences covering that whole tree of life, both plants and animals. And that's where, in part, where the GAP project comes in, because it's going to help deliver on that strategic action. And within that report, so it's available through the Australian Academy of Science, there's a whole raft of things in there that point to where we should be investing for genomics. But I guess the key thing is we want to try and accelerate our species discovery, and we see using some of the genomic tools that are available as a way of doing that. So to turn to plant systematics, taxonomy and systematic research, traditionally, like many areas, we've relied on a few markers, and with the majority of those being on the chloroplast, simply because they've been easier to get at, and a few markers because we've largely relied on Sanger sequencing. And Sanger sequencing is, still being used, although it's now clear that it's on its way out as a methodology, mainly due to expense and ease of getting greater coverage of genetic information. So the shift that we've seen really in plant systematics is a shift to whole chloroplast analysis. So still using the chloroplast, but there are challenges, because the assumption is that the chloroplast data parallels and is representative of the evolution of the species. And of course, the evolution of species is really a nuclear DNA question. And so how good an assumption is that? So I'll just run through a little example here to illustrate some of the pitfalls that we face in certain groups. So I'll pick a group that's very Australian, and this is the eucalyptus group, and in particular the ash species. So here we can see mountain ash, regnans, and a number of other closely related species, alpine ash and things that are in that same group. When we look at the chloroplast haplotypes, what we see is that there are haplotypes shared between the species, which sort of raises alarm bells if you're using chloroplast data to infer evolutionary relationships. And as you'll see, these things overlap broadly in their distributions, yet they have very different morphological characteristics. They have different altitudinal ranges. They're very distinctive in the field. So you can easily tell these species apart based on their morphology. And there are a couple of groups within these, the green ashes and the blue ashes. So yes, you've got that altitudinal replacement from as you go through altitude, the species shift and you get overlapping but quite distinct altitudinal bands in these things, which generally have sharp morphotype boundaries between them that are controlled by moisture and elevation, although hybrids do occur in those ecotones. So if we look at what the chloroplast data shows us, here's a phylogeny built on the chloroplast data, and we get nice resolution on branches, and we're seeing some geographic relationships in there. Lake Mountain and Mount Useful off to the left-hand side, and Dargo and Buchan off to the right-hand side, which more or less matches some of the patterns we see in species distribution. But when you look at the species you're dealing with, you'll see, based on the chloroplast data, that none of them are monophyletic. Flora scattered through the snow gums, the mountain ash is scattered right across it. So if those morphological characteristics that we use to identify species are valid, then there are some issues here, particularly in terms of using the chloroplast data. And basically what we see in a number of groups is chloroplast capture through hybridisation events and then back crossing to that particular species, but the chloroplast, because it's uniparentally inherited, stays in the system and then effectively gets captured and populated throughout that community. And so in part, this is because those particular species have leaky breeding systems. Hybridisation is something that rampantly occurs. And so the reliance on chloroplast markers in these sorts of systems is problematic. Now, it's not just eucalypts that this happens in. We're seeing this in a number of groups, number of Australian groups where chloroplast capture is happening, and that's possibly misleading us in evolutionary relationships. If breeding systems are tight and you don't get hybridisation, yes, the chloroplast marker is a good one, but given the shift to NGS technologies, maybe we should be thinking about shifting our strategy and focusing on nuclear markers, which are the reflection, or should have the reflection of that evolutionary relationship. So there are some lessons for the community. You know, we're definitely moving away from Sanger sequencing. We need to rethink what our strategy is in terms of doing these types of studies. Should we rely on the old approach based on chloroplasts, or should we shift onto something else? And so these are sorts of questions that GAP is addressing. So to turn to the project itself, Genomics for Australian Plants has three focus areas. It's reference genomes. So as I mentioned before, the genomic coverage across the tree of life is very biased towards certain groups, which means other groups aren't covered, and I'll touch on that when I talk about the reference genomes. There's a focus area on phylogenomics in terms of understanding, building those evolutionary relationships. And then there's a third focus area on conservation genetics, looking at how we might use genomic data to better target conservation strategies amongst the Australian flora. Cross-cutting all those focus areas is a capacity building element to upskill our community. Within that community, there is limited, or has been limited, bioinformatics capacity and capability. In fact, it's probably only a couple of years ago, maybe 2 years ago, that the first bioinformaticians were employed within the plant collection community. So there's a whole lot of upskilling and capacity building that's needed as well, and so that's another focus area of the program, to build that capacity within the community. And you will have seen at the start of the presentation the link to the website of Genomics for Australian Plants. So this program, like many other Bioplatforms Australia programs, has been built through wide consultation, trying to identify collective goals that people want to try and achieve, and then shaping those into a program. And so currently there are a large number of collaborators spread across the country. They vary from research institutions like ours that hold Herbaria, Botanic Gardens, and university sector as well, government departments and increased capability. So it's a really great group of people to work with, and what we've done in order to deliver it is we've developed the governance model where the steering committee is supporting working groups is identifying supporting working groups. Those working groups develop the scope and make recommendations to the steering committee for endorsement. And that's the governance mechanism. There's also some small financial contributions from various botanic various botanic gardens, CSIRO, Department of Biodiversity, Conservation and Attractions in WA, where we're putting in small annual contributions, and we're asking all consortium members to do that because the Bioplatforms Australia funding covers the sequencing effort, but it doesn't cover project support and a little bit of bioinformatics, but it doesn't cover the real needs of it. And I guess one of the things that we did recognise early on was the need to be able to bring in bioinformatics support for this project, which was greater than what Bioplatforms was able to support. So I've secured 2 grants, one through the Ian Potter Foundation and one through our Gardens Foundation, which is funding 2 bioinformatics positions for support, training and pipeline development. The steering committee is a mixture of skill sets and institutions. So we've got a really good balance here in terms of NCRIS, views from NCRIS and project management from the herbarium community, from CSIRO, from the university sector as well, and botanic gardens. So it gives us a good perspective across the whole community about where to go, and it also has got expertise. You know, as I mentioned at the start, I'm certainly no molecular biologist, but we've got people on the committee that have got expertise in bioinformatics and in genomics and in conservation that give us those important insights that we need to make the decisions. And the working groups are really where everything happens, and you know that's been the critical thing, to get that buy-in from the working groups. And really there's been a lot of energy invested by those working groups, both initially when we're working through, you know, what sort of laboratory approaches will we take, what sort of bioinformatics did we need to look at, and computational work, and then just on terms of the project development and delivery, the phylogenomics and the conservation genetics. So where are we at now? Well, we had an initial call for genomes, for reference genomes, just to really to test the water and get a better understanding of what was going to lead to success and what things we needed to keep our eye on. And so 3 initial genomes were selected to be sequenced. It's worth noting, I guess, acacia, which is one of the largest genera in Australia with over 1,000 species. So remember, we're talking 25,000 species in total, and 1,000 of those are acacia. There is no genome for acacia. We selected tilapia, which is within the Proteaceae, and again, that's a really important Australian family. There are over 1,500 species in the family globally, and the majority of them are in Australia. And again, macadamia, which is a crop plant, is the only genome within that family. And both of those groups are, you know, long-lived, or both those taxa that we've selected are long-lived and woody, and provide the opportunity, I guess, for us to have a living accession. So there'll be a voucher accession in a herbarium, but a living accession to go back to if other studies want to be done. And I think that's an important concept within the project, trying to build long-term opportunities. And the third genome is a thing called Aerocleome, which is in the brassicale. So, you know, people will be well familiar with genomes in that group, but here's an Australian one that hasn't been sequenced, and I'll come back to those a little bit later at the end. In terms of the phylogenomics, there is a global project run out of Kew called Pathtoll, Plant and Fungal Trees of Life, and they're aiming to sequence all genera of flowering plants and a bit over 8,000 fungal genera. And for the flowering plants, they've developed a bait set, which has 353 nuclear markers in. So we've been talking with them, and we've developed an MOU with Pathtoll to help both parties achieve our objective. So they're going to be sequencing, well, they've already sequenced some Australian genera. We're going to fill in the gap with the rest. And our vision is that we'll have a data set that covers all vascular plant species in Australia, an ambitious goal, the 24 or 25,000 species to sequence, and then analyse and put into a phylogenetic framework. But we're starting off small with the target of doing all the genera, which is about 2,109 at the moment. So the other piece that I mentioned at the start was the capacity building and training, given the shift that we're going to see in sequencing methodologies, or the shift that we've already seen, how to upskill our community quickly and effectively. And so Anna Symes, so I'm at the Gardens here, is leading this effort, and so we've already got a portal up. Pages are still under development, and we're planning face-to-face, or we're going to have a face-to-face meeting in the middle of this year, but that's somewhat been derailed by the pandemic, so that'll be delayed, but planning sort of face-to-face and virtual training to help students and postdocs that are working in this space, and also established researchers, and trying to give them a space where they can go and learn about genomics and upskill in that area. In terms of the pilot reference genomes, there's been some really good progress, and one of them we've actually put on pause, which has also been some great learning. So the aerocleome, which is on pause, is an annual effectively. And so how do you maintain a lineage of that? And we've had issues with tissue culture and just getting enough high quality DNA generated to be able to do it, and it's just proved extremely difficult. So we've actually pulled the pin on that one for the moment, but we've actually learned some things along the way. The acacia genome is probably the most advanced at the moment. So we've used MinION and ProMethION ion runs. We've got 10x and Illumina short read, and we're trialling different assembly approaches at the moment. Anna's working on the plastome and mitochondrial assembly, and we're currently in the process of doing a transcriptome run to help with the annotation. The tilapia one has taken a slightly different approach. They've got chromium 10x and nanopore, and they're aiming to add in high C to help with the scaffold assembly. So we'll have a couple of different approaches here and see which one's the most effective, and that'll help us with the next round of EOIs, which is about to be issued for some more genomes. The phylogenomics, as I said, is settled on the angiosperm 353 bait set, and also looking at using a bespoke bait set developed in Adelaide by Michelle Waycott. And we'll run a pilot or run a test to compare between them, same samples, and look at how both bait sets perform. Where we've had an EOI out for material, and we've got people contributing over 90% of the genera that we targeted, which is pretty amazing because we thought we'd struggle to get it to 60%. So it's been really very pleasing, and we're currently identifying about the 10% orphan genera and seeing where these could be sourced from. So we're getting close to running or starting to run that material. And the Conservation Genomics Working Group have worked through an agreed approach, and the EI is just about to be released, and the focus of that's really going to be about resolving species concepts that we know contain conservation dependent entities, and then that'll give us a better understanding of distribution of those entities and being able to target conservation effort. As with all of these NCRIS type of projects, the data will be publicly accessible, and there's sort of a 12-month mediated access period while we, focus on getting some initial things out, but then they will become available. And I guess many of the outcomes, the activities that are within this program are designed to deliver on the Decadal Plan for plant taxonomy and systematics. So we're not just upskilling our community, but we're creating plant genomic resources that can be used. And by tying it back to vouchers, which is sort of one of the key bits, that information can be fed into, spatial analysis and a range of other things. So I'd like to sort of close off quickly. This is a list of the partners that we've currently got, and acknowledgements and people that have helped with this presentation, and of course the supporting organisations that are making financial contributions are listed here. So thanks very much for listening. I hope you've got a better understanding of the Genomics for Australian Plants program, and I'll throw it over to Christina for questions. Thank you.
All right, thanks very much, David. It's been a wonderful overview of the efforts to untangle plant genomics data, both by collaborative international projects and also, importantly, by Australia's own GAP. So now's the time for questions. If anyone in the audience would like David to answer a particular query, now's the time to put it into the Q&A box on the dashboard. David, you mentioned too that upskilling the community in bioinformatics was an important goal, and the Australian Biocommons is working closely with GAP bioinformaticians, particularly in the area of de novo genome assembly and annotation. And we're very pleased that Dr. Anna Syme, who you mentioned, has agreed to deliver a webinar on chloroplast genome assembly using Galaxy Australia later on in the year. So we'd encourage everyone to sign up to that on our website too. So just while we're waiting for questions, David, I wonder if you might be able to give us a quick general update. No doubt the Royal Botanic Gardens has faced some very difficult times with COVID-19. And can you tell us what's going on in the gardens at the moment?
Well, like most other public places, we're shut to the public at the moment, but that means that we've got the opportunity to do a whole lot of maintenance and work at the back. And I guess, while I run the herbarium component, which has got the dried material, the living collections are equally as important, in Melbourne. And for those of you that are based in Melbourne and know the Melbourne Gardens, it's has the appearance of a European landscape, but of the 8,000 odd species that we grow, more than half of them are Australian. So it's sort of something that really surprises people when they hear that. And because I guess Botanic Gardens maintain curated collections, that is provenance collections of plants, those plants provide opportunities for this sort of program. So, we'd be looking at, sequencing living things that are going to stay in the gardens for a long period of time, so that if someone wants to come along and do some other omics studies on them, be them transcriptomes or something else, we'll have the genomes assembled for those things, or the bait sets done for them. So I think that's sort of one of the opportunities that we're trying to create here, is sort of build those long-term data sets, which of course is something that Herbaria are being good at over time.
We've had a question come in as well about raw data, and to what degree the data is available for independent analysis, or you know, non-partners using the data that's available.
Yeah, so all Bioplatforms projects have an open data policy. That data is all released after 12 months. So we'd expect all the raw reads sets to go up, together with, you know, scaffolds and annotations and alignments and all the other bits and pieces that flow from those raw reads. So yes, they will be available and openly accessible through the data portal. So at the moment, you need to log into the data portal, and that's available to consortium members, but after the 12 months, they become open access. So I hope that helps, Stuart. Okay.
Well, I think you've given a very comprehensive overview. That's the only question that we have for you today. So I think from that point, we'll wrap it up. And so once again, I'd like to express our thanks to you, David, for finding the time in this very busy period to share an overview of the Genomics for Australian Plants project. Thank you.
Thanks very much, Christina. It's been a pleasure, and hopefully everyone's got something out of it.
I'm sure they have, thanks. We'd like to acknowledge our primary funders. The Australian Biocommons is enabled by NCRIS via Bioplatforms Australia funding. Thanks so much for watching, and we hope you found it useful. Until next time, goodbye.